All right, welcome everyone to our Build a Cell seminar. Thank you for all the guests who are here, and thank you, Aurélien, who will be presenting their research. Very excited to hear about your work. It sounds really cool, and I'm loving this. So take it away. Thank you.
Thank you. So, I'm supposed to say a few words about my background and who I am. So I was trained in France, even though I'm working in Switzerland, in Geneva. Mostly, I've been doing membrane reconstitution assay and study the biophysics of lipid membranes with pure proteins to understand remodeling of membranes by proteins. But what I'm going to talk today about is completely different, is still reconstitution. We're trying to understand how morphogenesis can be produced in vitro using a few types of assays. That's what I'm going to tell you about, today. And that's the title is Topological Defects Drive Morphogenesis of Cellular Tornadoes and Bicephalous Hydra. So it sounds a bit weird, but you'll see where we're going. So to click, I guess, yes, here. So the story starts by something that you most probably all have seen in nature, looking at organisms, whether they're plants, whether they're animals, whether they're fungi. Usually the body plan of organisms is composed of order and disorder. So you have lines of order, and you can see on this nice Romanesco cauliflower that you have lines in spirals linking spots of growth, which are in this case the stems and the growing bodies of the cauliflower. But you have joining all these lines of order, you have one specific defect. There's many actually on this type of organism, but there's one central one, which is where all the lines are connected and where you actually lose the order, and that's the main stem. And so if you look in other organisms, and this is the example of the sea urchin, you also see that you have lines of order, which in this case are the positions of the spines. And you have the sensory organ on the top, and you have the mouth on the other side. So you have actually two defects, which connects the lines and where you lose the order. And you can also find this on yourself. It's a bit more subtle, but you probably know that on your head you have a defect connecting your hair and on their fingerprints. And essentially those defects are linked to several aspects of the body plan. One of them being topology, it's just because you are functionally speaking a sphere, that you will necessarily have two defects like you have on any sphere. If you have in other places you have order. That's the starting point. So of course the question you may ask is, if there are topological defects at specific places in the body plan of organisms, is that meaning that they have a role during the building of the body plan, which is actually morphogenesis? And if yes, the question is, what could be their role in morphogenesis? So now we can look into small animals and also at the cellular level to see what could be their role and what kind of defects we can find in those simpler model systems. So one of these systems, and I'm going to talk about later in the talk, is the Hydra. And the Hydra is a very simple animal that you probably know. It's a diploblastic, so it has two layers of cells. I'm going to come back to this later. And if you look the ectodermal layer and you look the actin structure in the ectodermal layer, which is the skin of the animal, you find this beautiful order of actin fibers. They're all aligned like a nematic, very nicely from the foot to the head. And if you look at the position of the mouth, and this is probably here that you see it the best, you have an aster, and all the actin connects or points towards the mouth, and you lose the order at the mouth. So it's a defect. It's called the +1 defect. I'm going to come back to those numbers and explain what they are. It's called the charge of defects. And essentially, you see that the animal, if you accept the tentacles, has the topology of a sphere. It has 2 + 1 defects, or 2 poles, one at the foot and one at the mouth, and these poles are connected by actin fibers, essentially. So that's very interesting, because these fibers are, functionally speaking, the muscles of the animal, and it's very well described that through waves of contraction, they participate in elongating the tissue, notably during regeneration, and so they create the force that is required to shape the tissue, essentially. Now, what you can do is also take myoblasts, which are cells that come from the muscles, or muscle precursors, I should say, and you can plate them onto a petri dish, as you see here. And because they are muscle-like cells, they're very elongated, and they're very contractile. And so those cells, which are C2, C12 cells for people working in the field of muscle and muscle regeneration, these C2, C12 cells, they form a very nice nematic order, which is this alignment of cells that you see in green in this box. And in the field of orientation that you can measure here, so the nematic order, you find defects of 2 kinds, the red ones and the blue ones. And so these defects are theoretically represented on the right part of the slide, and they are associated with 2 numbers, the +1/2 and the -1/2. And these numbers are called the charge of the defects. And you can see that they correspond to a specific pattern of orientation around the defect being in the center. And to explain those numbers, it's fairly easy to understand. If you take the vector of orientation of the cells in whatever position, and now you look how this vector will change during a rotation around the defect, you will see a different behavior. You see from the red example, which is the +1/2, that when you spin left, the vector is also spinning left, but makes half a turn when you do a full turn around the defect. So the plus tells you that the vector is turning the same way than you, and the -1/2, the 1/2 tells you that the vector is doing half a turn when you're doing a full turn. So you can do it also with the blue. And with the blue, you can see that the vector is actually spinning right when you go left, but it still makes half a turn when you do a full turn. So that's a -1/2 defect. So these are the only 2 kinds of defects you can find in cells, the C2, C12 in particular, but also all the cells that display a nematic order in vitro. What is interesting is that these cells are contractile. And if you now look the stress pattern associated with the defect, you can find a very reproducible pattern of contraction. So this is, you see the specific organization or order of cells around the +1/2 defect. And you have measured by traction force microscopy the stresses generated by the cells around this defect. The blue one is showing extensile stresses, and the red one are the compressive stresses. And you can see that mostly around defects of +1/2 and -1/2 charges, you find compressive stresses. What is very interesting in this paper from the Ladoux group is that the position of these defects has been linked to a phenomenon specific to cell monolayer, which is extrusion. So the fact that cells will be pushed out from the cell monolayer to reduce the number of cells, and specifically it is associated with the position of defect. You have approximately a +50% increase in probability of extrusion at the defect position. Why is that interesting? Is because you have a stress pattern that is compressive at the defect and probably pushes the cells up. So that's interesting, because you can connect the stress pattern generated by the contractility of cells and their specific organization around the defect, and a phenomenon that depends on the force, the compressive stresses, which is extrusion. So that gives you a hint about what is the hypothesis that we came up with, which is that maybe some, maybe not every, but some of the topological defects can organize the stresses of contractile cells in the tissue to shake the tissue, so to generate the forces needed for morphogenesis. So that was the starting point that we imagined for the study. So Pao Ahmad was a brilliant postdoc in the group, and he started with this idea by designing an assay where he could control the localization and the charge of the defects, and concentrate the stresses enough so that instead of seeing, for example, extrusion, he would see deformation of the entire tissue, essentially. So to do that, he used a very simple technique. And this technique is micropatterning. You essentially cover the surface with a repellent to cell adhesion, and then you burn some of the places with a specific pattern to actually make the cell adhere only to some parts. And in this case, he chose discs of certain sizes. You can change the size. And if you now plate the cells on those discs, as you see from this picture, you again display a nice nematic order with defects, and the defects are the same than the ones I've told you before, which are the -1/2 and the +1/2. The total charge of the disk being +1, meaning that you need to have a total charge of defects, the topology of the disk being +1, that means the total charge of the defects is actually +1. So if you reduce the size of the disk, you reduce the number of defects, but their total sum will still be +1. As you can see from here, there's only left 2 +1/2 defects, and so it's the total charge is +1. And if you even reduce further the size, you will start organizing those defects in specific places, so you can have even more constraints. So the idea of Pao was fairly simple. The idea was, can we reduce the size enough so that those 2 +1/2 defects would be fusing together to form a +1 defect? And why +1 defects are interesting? First, because there are several classes of +1 defects. You have concentric rings, asters, and spirals, anything that is in between. And as you can see from the specific pattern displayed around the +1 defect, there are isotropic defects. So that means in every direction, you will have the same organization of cells. And that leads to higher concentration of the stress. So the idea was, can we generate +1 defect to generate high stresses in those flat monolayer on disks? Indeed, Pao was able to do that, as you can see from this movie, where the C2C12 cells onto the pattern were spontaneously organizing as a rotating spirals at the beginning, and then later you see a transition with something that looks more like an aster, where you see all the flows coming in and an accumulation of cells in the center. Also, if you look the angular speed with time, you see that it goes up when the spiral is forming, and then it goes back to 0 when the spiral transition to an aster configuration. We get the spiral at low density, and the cells, as you have seen on the movie, are proliferating by just having a lot of cell division. So the density increases, and the spiral transition to an aster. So we get the aster only at high density on these movies. When we show this transition to our colleague in Geneva, Carsten Kroes and Carles Blanche-Mercader, they got very interested because Carsten had predicted a transition from spirals to asters from an active gel theory, in this case, nematic gel theory, by just changing the active term, so the energy spent into activity, contractility of the whole gel. And you can see that this is in this publication that they predicted a transition from spirals to aster when you increase the value of the activity. For reasons that I want to explain today, we needed a theory a bit more general than what was in this paper, and Carles actually did the theory, and you can read it in the paper highlighted in blue there. And what makes this theory more general is that it takes into account the two modes of activity in those active gel nematic. The first one is what is called directional motility, or just the fact that elements can move with a certain persistence in their movement on a certain distance. So essentially it's motility. And the second one is what is called anisotropic contractility, the fact that the element can contract in different directions with different forces. So that, for example, this is one case where the cell will push in one direction and contract in the other one, but you could imagine that it simply contracts in every direction, but with different intensities. So by doing this, you can predict the stress field of different configurations. And here you have 6 different cases. The column on the left is for asters, the column on the right is for spirals, and this is with one set of parameters. The second line is with a second set of parameters, and the third line is with a third set of parameters. If you look carefully, you see that with the first set of parameters, both the asters and the spirals are extensile. You see that I forgot to say that the arrows in purple are showing the stress field, so it's pointing outwards. Whereas with the second set of parameters, you see that the asters are contractile and the spirals are extensile. And the last one predicts contractile. So that's with the third set of parameters, you see that both the spirals and the asters are contractile. And that's actually with the experiments we have been doing, the set of parameters that corresponds to the experiment is actually this, predicting a contractile stress fields in those structures. So is that the case? Is that something we see in the experiment? Well, the first thing we see is that these, after 5 days of growth, these flat monolayers grow into dome-like structure or mounts, if you wish. And maybe it's more easy to see it by having this higher magnification. These mounts keep a certain structure and a certain order in them. It's not like they become completely disorganized. And I want you to focus maybe on the picture because this is more easy to see. You see here the organization of cells through actin labeling, and you see that at the base of the mount, and these are pictures going up into the mount and single confocal slides, you see that at the base you have an aster configuration with all the fibers pointing towards the center. And if you move up, you have a transition towards a spiral, and at the tip you see a spiral. This is probably also visible in this. This is the 3D vector field of order in many mounts, average over many mounts. And if you see the average vector at the right, at the bottom, the purple one is aligned with the radial direction, whereas at the top it's the blue one, and you see that there's an angle corresponding to a spiral. Now, what you can do is also segment the surface of the mount and project the fluorescence of the neighboring cells onto the surface and look at from the top. And so here, this is what you see here. This is essentially a picture of the cells on the top of the mount, not all the cells. And you see that these cells are very well organized as a spiral. What we can also do is measure or count the number of nuclei on the basal layer and on the top layer of the mounts by segmentation again. And you can see from the plot on the right that for each mount, there's a lower density of cells at the top than at the bottom. And so that's interesting because it's exactly the opposite transition than what I've showed you before. What I've showed you before is that through proliferation, the cell layer gets more dense and transition from a spiral organization to an aster configuration. Here, it's just the opposite. Cells are being pushed from the monolayer to the top, so they decondense on the top of the mount, and they transition back to a spiral. So they again, the transition is occurring through a change of density, but it's the reverse change of density, meaning that you go from an aster configuration to a spiral configuration. Not only those spirals are visible at the top, but they do spin, as you see from this movie, and the cells are moving on the top of the mount. So we get structures where we have a specific order that is linked to the density. So the proliferation triggers the transition from spirals to asters. The growth induces the formation of mounts, and they have a spiral configuration on the top and an aster configuration on the bottom. And so, structurally speaking, these mounts have topological defects on the top and on the bottom, and the defects are always +1 defects. And we think that they participate in organizing the stress field within the structure to generate those mount structure. The question remains, what is the stress field in the mounts? Because of course, all is based on forces, at least our hypothesis. So we needed a way to address the force field. So Pau came with the smart idea of putting a contractile pillar, so an elastic pillar, I should say, in the center of the disc. And as you can see from the movies, these pillars get constricted when the mount is forming around them. And I hope you see that the smallest pillar actually gets constricted more than the other ones, meaning that you have more forces in the center of the disc than outside or on the border. And so if you plot the max compressive stress as a function of the pillar radius, which means depending on the radius in the disc, you see a force gradient with higher forces in the center and lower forces onto the side. So that shows that you have a force gradient, and that shows that this force gradient probably structures the entire mount form or shape. Now we wanted to know whether these structures could actually do essentially the two things that you can see in morphogenesis, one of them being differentiation, so cells taking another step in their cell fate acquisition. And the second question was, could we go further than just having mounts? Could we get shapes that were much more elongated, for example, or much bigger? So one of the interesting things about the C2, C12 cells that we have used is that they are intrinsically able to differentiate, and they do that if you stress them, meaning mechanically stress them. They like being stressed like any muscle, and this differentiation process is being trackable by looking at the overexpression of myosin heavy chain, which is specific to the muscle formation. So if you look where those cells, so the differentiated cells appear in the mount structure, what you see is that they are mostly located in the center. This is a single picture, and this is an average of 43 different patterns, and you see that the green is mostly in the center. And that means that where you have the highest stress, you have more differentiation, which is expected from what was known before, because those cells differentiate under stress. The other interesting thing is that these cells can actually lose their ability to differentiate after many, many passages. And you can see the difference here. This is essentially the same line, but on the right, you have what has been passed, I guess, more than 60 times, and the cells have lost their ability to differentiate. And on the left, you have the normal cell line where you see plenty of differentiation after 3 days of in vitro culture. Why is that interesting? Is because you know that in morphogenesis, there's usually a competition between differentiation and proliferation. That's why we have stem cells. The stem cells keep on proliferating and have no differentiation, but the cells that are being the daughter of stem cells, at least one of them, usually acquires a specific cell fate, differentiate, and stops proliferating. So we wanted to know what happens when the cells cannot differentiate anymore and keeps on proliferating all the time. And so in this case, you can start to see structures directly by eye, looking at the bottom of the Petri dish where the cells are being cultured on the discs. They look like this by bright field, and they look like this by confocal microscopy. And so they acquire a very elongated shape. You see that the growth is constrained to the central part of the disc and form these amazing towers, protrusions. And these protrusions actually seem to have a spiral organization on the top with a defect, and the cells are organized as a vortex around the core of the structure. If you change the boundary conditions by just changing the shape of the pattern from a disk to a square or to a triangle, you see the same phenomenon. That is because it's just linked to the topology of the surface, and the topology of the surface in all these cases is +1. So as long as you have a +1 defect in the center, you can get the tower. And not only they look like spirals or vortices, but they spin, and they spin. This is one seen from the top, and you see that it rotates. And that means really what they are is tornadoes, because they essentially behave like tornadoes. The motility of cells, the collective motion of cells into a vortex, driving the constriction and the growth into one direction. You see also at the end of this movie that we lose the repellent agent on the surface, and so the cells are crawling out the structure and everything falls down. So you lose, when you lose the confinement that sets the topology value to +1, you lose the structure. So it's really the +1 defect that is the driving condition or constraint to form those towers. So they can get very long, and they get, this is structures that are about half a millimeter, and they keep this twisted shape that are reminiscent of the tornado effect, I would say. And you can see that they form very large protrusions. So we think we have a fairly simple mechanisms of growth here, where the cells initially organize as a spiral onto a monolayer, and through proliferation transition to an aster, and the aster is actually more contractile, so it pushes the cells towards the center, forms the mount, and the mount top reverts to a spiral configuration that grows into a vortex when the cells are kept under proliferation. So we think it's a very simple mechanism of morphogenesis, combining proliferation and order, and of course motility and contractility. And at least we know that there's one organism that does that. It's the slug of Dictyostelium, the amoeba, where one of the stages of development is actually linked to displacement of a slug, and this slug is actually a rotating tornado and looks very much like those protrusions. So I'm gonna switch back to the Hydra that I presented rapidly before, and this is the work of Yamini Ramachandran in the lab, and you will see how it is connected directly to what I've showed you before. One of the points being that in the Hydra, as I've said before, we see a very nice actin structure. So Hydra is a model organism actually that is directly linked to Geneva. The reason being that you all know the regenerative properties of the Hydra. If you cut it into pieces, it regenerates the missing part. If you take the foot tissue, it regenerates the head, and the head will regenerate the foot, essentially. It was these amazing properties were discovered by Abraham Tremblay, who was actually a zoologist in Geneva a long time ago. And he discovered the Hydra, and he discovered the amazing properties of the Hydra for regeneration. And that was one of the founder. He was one of the founder of experimental zoology by doing this experiment. And all what I'm going to show is a collaboration with Brigitte and Matthias from the biology here in Geneva. So I've told you that there's an amazing nematic order of the actin fibers in the ectoderm, and that's a longitudinal organization. That's the green pattern that you see here. But in the endoderm, which is the second layer of cells, you also have a very amazing actin organization, but it's circumferential. So it's 90 degrees from the ectoderm organization. I'm just going to show you the ectoderm, which essentially corresponds to that kind of picture. But just to mention that there's two layers of muscles which are 90 degrees apart and probably contracting in opposite directions. Not opposite, 90 degrees apart. What I'm going to show you is the regeneration of the head from the foot tissue. So if you take a Hydra and cut it in 3 pieces, you can look at the regeneration of the foot from the head. The central part will be able to regenerate both the head and the foot, and the foot will regenerate the head. So I'm going to show you only head regeneration. What Yamini did was to look at the regeneration process under the microscope. And usually to do that, you embed the tissue into agar. That's the control experiment. And still, you see that the animal is moving a lot by contraction along its axis. And during this regenerative process, it forms a head. You see the tentacles coming out, and you will see that it grows a longer term. After a while, I have to say, it moves so much that it becomes difficult to image. And that was one of the reasons why she tried to, instead of embedding, do agar compression. So she took the animal and put a slab of agar on top of the animal, compressing it a little bit. And in this case, she observed a striking phenotype, which is the formation of 2 heads. And you see it here. And you see very well also the actin organization in this movie. And you see the 2 heads forming, and it's really an axis duplication. You see that there's 1 foot and 2 heads. And that was actually never seen before. If the slab is too rigid, instead of regenerating, the animal sees its cells being dissociated, and the animal dies from dissociation, essentially. These are the statistics. And you see that in the control, we never, and that's known, so that there's not as much statistic in this case than in other cases. It regenerates a single head from a foot tissue. If you use 0.5% of agar, you see that there's a substantial amount of animals that grow 2 heads. We have also ectopic tentacles and a little bit that does not regenerate. If you increase the agar concentration to 1%, you see actually a higher proportion that do not regenerate, and I'll come back to that. A small proportion that dies, and then all the other proportion are reduced, essentially. And at 2%, then death becomes the majority. You kill the animals by dissociation, essentially. So this is to show you that you can also release the animals after compression. They keep on growing, and after a few days, they look like very normal animals. They have a head. You can see that by a Wnt RNA hybridization technique, and they have a single foot in the control. That's the red one, which is staining for cream, a molecule that causes adhesion of the foot to the substrate. But after compression, the 2-headed animals have really 2 Wnt signaling sources which are linked to the head, and a single foot. So they really are 2-headed animals. And not only these animals are, have all the features of adult animals, they also feed very well, and the 2 heads are able to grab the shrimps and eat normally, which is a functional test to the animal. So we have functional animals at the end. So the model is very easy to understand. What we think happens is that in the foot tissue, we keep a single defect, which is the foot. And so the actin is oriented like this. And the regeneration starts by actually wound healing of the top, forming another defect from where the head will grow. What happens when you compress is probably that we get, instead of a single point defect, we get a line defect, and this line defect is not very stable and splits into 2 or more defects on the animal once you have done the wound healing. And these multiple defects create multiple heads. So under compression, the idea would be that we form several defects, and that's what forms the head. Of course, there's an assumption here that I don't really say, which is that you need to have one defect to regenerate one head, so that's the defect is needed and mandatory for growing a head. And this is something that is not shown because this is correlative for the moment. I still want to mention that we still think that we have created the mythological Hydra, because as you may remember from the old books of Greek mythology, Heracles was able to kill the Hydra, but each time he was cutting a head off, there was several, or at least two heads growing. So in this case, from the real Hydra, the one that we found in our ponds, we think we have created an animal that looks much better and much like the mythological Hydra. Anyway, the important point is that compression usually occurs laterally, like what I've shown you before, which is that the tissue being a little bit elongated, it falls on the side when we put the slab, and the slab laterally compress it to form the two heads. But in some cases, we are able to compress it vertically, and we can compress it vertically with the foot being on the bottom or with the foot being on the top. And in this case, we could find a very different behavior. This is seen from the foot being on the bottom, so looking towards the microscope. And you can see the beautiful organization of the actin into asters. So the defect is in the center. And you see the animal tries to contract to regenerate, but forms actually holes or tears up the tissue in the center and forms two holes that actually fuse together to form a real hole. So in this case, we generate a toroid animal. It fuses back on itself and makes a hole in the center. So why is that interesting? Is because the topology of the tor, the total topology of the tor is zero actually. So on the sphere, if you look the Höller number, you find +2, and that's why you have 2 poles on the sphere. But on the tor, you actually have the Höller characteristics of 0. That means it's a surface that in theory, I mean not in theory, can have no defects. Of course, you can have defects, but the total sum of defects will be 0. And so that means you can even have surfaces with absolutely no defects. And of course, if you have no defect, that means the lines along the surface will be organized like this, meaning that some of them are circumferential to the tube structure, and some of them will be longitudinal to the tube structure. And in this case, you have no defect. So we wanted to know whether the actin maps in our animals were looking like a no defect structure. And indeed, if you look with a very simple orientation G tool, you see that mostly the actin in this case is organized perpendicularly to the axis of the tube. But we wanted to go a bit further, so what we did was to use light sheet microscopy to look in every direction. This is a single animal, but look in different angles to really check that there was no defect. And from this picture, our conclusion was that there doesn't seem to have defects. Now we can take those animals and look at them for a certain time, which is in this case 72 hours after release from compression, to see how they behave. And you can see that they contract. You see the lumen of within the tube, actually within the tor, but you see that they don't regenerate heads and foot. And at the end, they contract and are never able to regenerate. Essentially, they die from starvation after 3 or 4 days, which is expected because they don't have a mouth, so they cannot eat. So the conclusion of this experiment was, if you have no defects, you get no head. Now, is that a property just of the toroid, or whether it's a property of having really no defects? So we wanted to have a toroid with defects to see whether in this case we would generate a head and a foot. So in this case, we use a different tissue, and this is the spheroid regeneration. The spheroid being the middle part of the animal that closes back onto a sphere as soon as you have cut it off. And this fragment will regenerate in different ways. So we compress those tissues, and in this case, the regeneration starts with a disorganization of the actin. And in this case, if you generate a toroid by compression, you actually can form a head and a foot, as you can see from here. So you see the tissue tearing and the opening, and you see that the foot and the head are still forming. The foot is on the left, I think, and the head will regenerate somewhere here. Okay, now you start to see the tentacles. Okay. What we did was to look at the actin organization in those toroid animals. And what you can see is that in some cases, we found the same organization of actin than in the previous toroid that was perfect, where the actin is oriented at 90 degrees from the axis. But in some cases, in some other places, the actin looks completely different. And I hope from the right one, you can see that there's a defect actually here and a second defect here where the foot is going to happen. So you can generate toroids with defects, and in this case, they regenerate a head and a foot. And these are the defects. I forgot that I had these arrows to show them. And these animals actually live also well after being released. They have of course, they have a hole in their belly, essentially, but they have a single foot and a single head, and the head is functional because it grabs the shrimps and eat the shrimps very well. You see very well the hole in the belly in this case. Okay, and my conclusion, and I'm going to stop here. Mechanical deformation of the regenerating hydra can create two heads. The actin nematic order forms two integer defects under compression, and we think that these defects are responsible for forming the two heads. And when you change the topology of the animal to a toroid so that it has no defect, no head is formed. And the toroid tissues with defects can regenerate a head and a foot. And so we think that the topological defects in the actin order are required for the head regeneration. And most likely it's because they set the stress field to form the body axis of the elongation that will then allow for full regeneration. So it's a bit of the same hypothesis than what I've showed you for the cellular tornadoes. And I'll stop here and highlight the names of Pao Guillamat, who did the cellular tornadoes work, and Yamini Ravichandran, who is doing the work on the Hydra. And I'll be happy to take any questions.
Yes, thank you so much. That was such a great talk. Very, very fun. There are already a couple questions in the chat. I'll again remind you to please read them out loud for anybody who's watching the recording later. And then welcome anybody else who wants to unmute themselves and ask their questions in real time, if you'd like to do that as well. But thank you one last time, Aurélien, before you answer these questions. It was an amazing talk. Sure, thank you. I'm going to start maybe with some of the questions being read on the chat. So I take the first one. Are those spirals and other shapes controlled by some particular known protein in the cytoskeleton? Could you influence it by mutation? That's actually a very good question. The structure themselves, not really, but the chirality of it, certainly. So these spirals are actually mostly counterclockwise. It's actually 76% counterclockwise. And for C2C12 cells, I mean, because it depends on the cell type you're going to be using. And this is set by the ratio between spectrin and actin in the cell. That's work from Sasha Vershatsky, I think in the Weizmann Institute in Israel. So it's pretty clear that you can regulate the chirality. We expect that the fact that you have spirals and asters may depend also on the contractility, because the theory points out the fact that if you have a higher activity, which means in this case a higher contractility, you should get more asters.
But it depends also on density, and that's something we are theoretically exploring, how density of cells could participate in changing the spiral to the aster configuration. Shall I go on with the questions? I see it's coming up, so maybe I continue reading. Second question, did anyone try reconstituting any of the Hydra actin and other cytoskeleton parts in vitro? It would be fantastic system for engineering cytoskeleton. So that's a good idea. I think one of the good idea, and what people have tried, I think, is just to cut off the tissue. So usually you have a cylindrical tissue. You can open it along its axis and then like this and put it on the substrate and look at the actin and how it changes. One of the issue is that actin is everywhere in the cell. What I've showed you is only what is close to the extracellular matrix, but there's lots of other structures on the apical side of cells, which is for the ectoderm outside the cell and for the endoderm inside the cell. And so it's not so easy. I mean, it's really when you focus the confocal slide onto those structures that you see these amazing structures, but you have plenty of other structures that participate in other functions of the cell. So it might be a bit difficult to use directly the cells. I've never heard of actin purification and full reconstitution in vitro, but you know, actin is in theory, you can purify actin from any kind of cells as long as you have large amount of the material. So it should be possible, and I see no problem in doing so. Third question. Awesome research. Thank you. Could you speculate how generalizable that work is to other morphology, especially in higher eukaryotes? Okay, so I can give you one example that is unpublished. I mean, you can find the paper on bioRxiv. It's a paper from our colleague in Geneva, Marcos González Gaetan, where they looked at the fin growth in the zebrafish. And the fin starts as a cylinder and then grows into this paddle structure, of course, to form the fin. And doing so, you see actually at the beginning that there's an aster of actin at the tip of the growing fin. And this aster transforms into a line. And during the transformation to a line, that's when the shape transforms from an isotropic shape to a paddle structure. And so we think that this could be a case where you have a defect, a punctual defect that helps the growth into a protrusion, but transforms into a line to acquire this paddle shape, essentially. Again, this is very speculative, but it could be something interesting. In general, I think looking at actin defects, which are actually predictive to the cell order in tissues, in developmental conditions, different types of organisms, and different steps of development, is probably a very nice project. That's my guess, but of course I'm biased. So next question. The spiral orientation reminds me of the shell of snails and even fossils. They have that set chirality too. Do you know it's controlled by some similar mechanism? So I kind of answered the question directly, indirectly. Yes, the answer is yes, there's chirality to the spiral structure. It's about 76% counterclockwise. And this is set by the ratio, at least that's what is known, by the ratio between spectrin and actin. So we think it's the main control. And I think there's a last question. What is the natural environment benefit of forming two spirals and other structures, those spirals and other structures? So for the spirals, it's totally, I would say, in an artificial environment, but I can only speculate on what could be the benefit. So first of all, I think it's an interesting collective property of the system to auto-organize. These growing spirals, the cellular tornadoes, are interesting also because they are bilayered, if you wish. This I didn't show. So the cylinder is a bulky tissue. It has cells inside. It's not just a surface. The cells around are organized as a spiral and they rotate like this. But inside, the cells that are inside are very elongated and very long, and they look very much like muscle fibers. So we think that the stress generated by the cells around could shape the tissue into the shape of a muscle fiber. So that's our hypothesis. Of course, nobody has shown that, but it could be that by growing those protrusions, you end up having a real muscle fiber in the center after a while. Not outside, because the cells would keep on undifferentiated and will keep on rotating, but inside there's a line of cells that could fuse and form a fiber. I think we're all done. Thank you.
Yeah, there's a lot of really great questions. I love it. You've
Yes, it was
clarified
nice.
so much for me. Thank you. I'm going to put one last call for questions if anyone wants to post it in the chat or unmute. But one final thank you so much for a spectacular talk, and I'm excited to go learn more about it on my own.
Thank you. You see the sun is gone in Europe, so I wish you a very nice day, and I hope to see you all one other time.
Yes. Have a good night. Thanks.
Bye.
Oops.